Thanks for the invitation to talk to your group, and sorry that the Zoom format is requiring slides rather than a talk per se. I'm Rob Knight. I direct the Center for Microbiome Innovation, and I'm a professor in pediatrics, bioengineering, and CSE at UC San Diego. And my lab does a lot of work on the human microbiome. What I'm going to talk about here is how we're using AI to understand the human microbiome and some of the stuff that we're doing specifically in COVID-19. In addition to what I'm talking about here, we're also doing a lot on things like the campus wastewater sampling, a program that you've probably been getting a lot of emails from the Chancellor about. And the self-administered COVID testing that students can now take advantage of, and various other related topics. So if you know about some of the other stuff that my lab's doing, feel free to ask questions about that at the end. Are the slides advancing okay? You should be seeing the second slide now. Yeah. I'm going to assume yes. Okay, so I'd like to begin by inviting you to consider what you saw when you looked in the mirror this morning as you were getting up and getting ready to start your day. And for myself, I saw an organism that's just 43% human, and not just because it was early and I hadn't had my coffee yet, but when we think of what makes up our body at the level of cells, each of us has about 30 trillion human cells, but about 39 trillion microbial ones. So we're outnumbered in terms of cell count by the microbial side of ourselves. And that's where that 43% human number comes from. Now, you might wonder, it's the 21st century, shouldn't we be thinking about this at the level of genes and DNA now? And let's think about that for a moment. So each of us has about 20,000 human genes, but amazingly, the size of our microbial gene catalog is from 2 to 20 million microbial genes. And so by that measure, counting up unique genes, we're at best 1% human. And you've probably heard about all the excitement about the human genome and using the human genome for precision medicine. But astonishingly, when we just look at the human side of ourselves genetically, we're ignoring 99% of our genes. And most shockingly, we're ignoring the 99% of our genes that we can change and potentially have control over during our lifetimes as we modify those microbes, because there's no way you're going to modify your human genome. So the vast majority of these genes and these cells are bacteria in the gut. And the gut microbiome affects all kinds of things all over the body. And one thing the gut microbiome impacts is drug response. So a wide range of drugs ranging from painkillers like acetaminophen to even the latest checkpoint inhibitors that are used for cancer. Whether they'll work for you, whether they're safe for you, whether they'll have toxic side effects, depends tremendously on your microbiome. As I mentioned, the gut is not Vegas, and that what happens in the gut doesn't stay in the gut. And the gut microbiome is important for a lot of phenomena throughout the body, including obesity, cardiovascular risk, pain, but even psychological and neuropsychiatric phenomena like major depressive disorder, multiple sclerosis, anxiety, and autism. And we're finding that the gut microbiome is particularly important for healthy aging, where we have a large-scale collaboration with IBM in that area that I'll talk more about later. And it's especially important that we understand this microbiome and its complex ecology, because we're undergoing a global microbial biodiversity crisis right now. And I'm sure you're familiar with Rachel Carson's work from the 1960s, where she documented in her book Silent Spring how chemicals like DDT that were being applied to agricultural systems to control individual insect pests had far-reaching impacts on the whole ecosystem, wiping out the birds so that there was no birdsong in the spring, leading it to literally be silent. More recently, my colleague Marty Blazer a few years ago sounded a similar alarm call for the microbial ecosystems in our gut, which we're ravaging through the overuse of antibiotics, but also through a wide range of other issues, including overuse of C-sections, low-fiber diets, and so on. And what's amazing is that over the 20th century, as one disease after another caused by a single pathogen, everything from measles to tuberculosis was brought under control during that period. At the same time, we see this massive explosion of chronic diseases, including including things, including things like multiple sclerosis, diabetes, Crohn's disease, and so forth. And what's most amazing is that in 2002, when these graphs were published in the New England Journal, none of those diseases was known to be linked in any way to the microbiome, whereas today we know that all 4 of those and dozens of others are not just associated with the microbiome in humans, but we can cause or cure analogs of those diseases in animal models, thus establishing causality. And what's amazing is that when we look at people living very traditional lifestyles, like the Hadza hunter-gatherers in Tanzania, in the Great Rift Valley of Africa, what we see is that in those communities, we see strong evidence for seasonal cycling, but also when we use techniques like principal coordinates analysis to reduce all the microbiome data, the first principal axis separates out all of those microbiomes from the least industrialized to the most industrialized societies, where people like the Hadza and the Yanomami and so forth have completely different microbiomes from those in Australia or the US or Canada or Ireland. And what we're seeing there is that this high diversity community that we've co-evolved with, a lot of the major groups of microbes are lost as you get more and more industrialized. And so entire phyla, like the spirochetes, we see disappearing in anyone who's taken up farming, let alone anyone who's eating processed food and living in air-conditioned spaces, whereas other microbes like the bifidobacteria and the verrucomicrobia start to take over. So it's almost as though we've taken this amazing, diverse inner rainforest of our gut environment and bulldozed it and turned it into a cityscape where only the pigeons and rats survive and not much else. So you might be wondering, does all this diversity in the microbiome really matter? And I told you about links between the microbiome and a range of different diseases. So the gut microbiome is associated with things you might expect, like inflammatory bowel disease, things that are a bit more surprising, like, say, cardiovascular disease, and things that are really surprising, like autism's, Parkinson's disease, Alzheimer's. And one thing we've worked on for about the last 15 years with a range of collaborators, including Jeff Gordon-Saab at WashU, is obesity. And today, for example, I can tell you with about 90% accuracy, using a random forest classifier trained on your gut microbiome, if you're lean or obese solely from the DNA of the microbes in your gut. So on the one hand, this is an awesome trick from a technical perspective. On the other hand, we don't think it has a lot of clinical or commercial potential as a test for obesity, because I bet you can tell which of these people is lean and which one is obese without doing any sequencing at all, right? But on the other hand, if you try to do that classification task, lean or obese, based on human genes, you can only do it with about 57% accuracy using every SNP ever identified by genome-wide association studies, whereas we can do it with about 90% accuracy for microbial genes using random forest classifiers in both cases. And this just gives you an idea of the underlying data. So this is the AUCs for 32 SNPs, although the models don't get much better with the thousands that have been available in more recent studies covering hundreds of thousands of subjects, like the one recently published in Nature. And you can see that's basically a straight line diagonal. Whereas as we vary what's called the OTU threshold, where we group the microbes into more detailed or less detailed categories, what you can see is that by using an 82% identity threshold on the sequences, even back in 2011, when Dan Knights, my graduate student at the time, who's now a faculty member at Minnesota and who introduced the concept of machine learning into the microbiome field, he was able to get 90% classifier accuracy by using a broad identity threshold. But I want to point out that the underlying data is incredibly noisy. So at 82% identity, we're basically grouping the microbes at the phylum level. And you can see the incredibly noisy data where the same row corresponds to the same subject at 2 different time points, where there's tremendous variation one time point to the next, even in the major groups of organisms. And so this really speaks to the ability of random forests to pull out patterns from noisy underlying microbiome data. And all of this is very much a big data problem. So amazingly, each teaspoon of your stool contains in its microbiome DNA the amount of information it would take a ton of DVDs to store. So you can think about that the next time you're taking a data dump, as it were. Now, a large part of what's contributing to our ability to read out all this data is improvements in sequencing technology. And at the end of 2007, my lab did an experiment that simultaneously gave us insight into hundreds of microbial communities, including those from the lungs of cystic fibrosis patients, rivers across the United States, air, and even the Guerrero Negros hypersaline microbial mat, still the most diverse microbial community that we know of on Earth. And in this overnight run of the new sequencing instrument, we had the 454 instrument. We were able to collect and organize all that data covering 500,000 sequences from hundreds of different microbial communities. And at the time, the Sanger facility down the hall from my lab in Boulder was still charging $8 per sequence to do sequencing of DNA. So if we'd collected these 500,000 sequences with the old technology, it would have cost $4 million. But on the then new technology in 2007, we did it for just 12,000, and we did it overnight rather than over a decade. And so this concept of barcoding, so introducing DNA sequence tags with Hamming or Golay error correcting codes, and then using them to split a single sequencing run over hundreds of different samples, and then associate the sequences with the samples by the barcode. That's what enabled things like the Earth Microbiome project, where we crowdsourced hundreds of thousands of microbial communities from scientists around the world and sequenced them. But perhaps what's most remarkable is that today, that amount of sequencing that would have cost $4 million in 2007, 12,000 in 2008, today you can do it for just 75 cents. And this is what's really opening up a lot of these possibilities. If it costs $4 million per sample, you're just not going to do it. Whereas if it's less than $1, it opens up all kinds of possibilities. But just having the data is a big problem. And so to illustrate this, if I just give you a pile of numbers to look at, it's very difficult to understand what's going on in the data set. And if I arrange them into a natural order, you still can't really see what's going on. You might think that the problem is the low precision, so there's only like 5 distinct values in there at the moment. But if I give you more precision on each value, that doesn't help you either, right? Having more precise readouts at each point, you still can't see the pattern. And what we need to do is we need to visualize it in a way that brings out the pattern. So here I'm just going to convert each of those into a grayscale pixel, and you can see immediately what's going on. But one problem and source of controversy in the microbiome field is that a lot of people are focused on taking one sample and trying to get more and more out of that sample with deeper sequencing, longer sequences, that kind of thing. And to switch gears in the analogy for a little, it's kind of like you're doing astronomy, but the only tool you have is spectroscopy, and you're just pointing at one star, getting a more and more detailed spectrum. And while that's scientifically useful, that's not how you're going to understand the universe and the whole sky, where you need a huge number of pixels that you're going to stitch together into the overall pattern. And that's exactly what we've seen over the last 15 years in my lab with sequencing, where we need to look at many samples together. So this is what's called an Otu Table from Chime, our microbial analysis software that we develop in my lab, where what you can see what you can see is each row is a different kind of bacteria, each column is a different sample, and there's just 9 samples in this table. And it's very difficult to see the patterns from the data in this form. But in a recent project, not with 9 samples, but with more like 900 samples, with Peter Dorrestein, we were able to show the value of spatial mapping and interpreting what's going on. So, what you can see is a LiDAR scan of Peter's office with 4 of us sitting around the table, and the scale, and then it took a few hours to swab all of the people and all of the surfaces, and then months to run a mass spec for all the chemicals and DNA sequencing for all the microbes. And the scale on these figures is that blue is the least of something, red is the most. And you can see this microbe, Synechococcus, lights up one of the people around the table very brightly, but not the rest of us. And if you know much microbiology, you might think, isn't that a marine microbe? And sure enough, you'd be right. What we're seeing here is that Peter swims in the sea more frequently than the rest of us, although he'd last done it 2 weeks before we did the sampling. And molecular data is even worse. So this is the step after the equivalent of the Otu table for molecular data, where what you're seeing is spectra clustered by this similarity. So these are fragmentation spectra out of what's called tandem mass spectrometry, where you take the molecules, you sort them by their mass to charge ratio, then you break them into little fragments, and then you figure out what the molecule was by weighing each of the fragments very carefully. And then these are clustered into a network where we draw an edge between 2 nodes, which represent 2 spectra if they exceed a certain similarity score, typically with the cosine measure. But again, mapping them onto the spatial context of Peter's office, you can see one molecule that lights 3 of us up very brightly and very specifically on our faces and hands, but doesn't touch the 4th at all. And the molecule that we're looking at happens to be caffeine, and 3 of us drink a lot of coffee or tea, whereas the fourth abstains from them completely, although you can see that someone has spilled some on his floor at some point in the past. And while right now this is undoubtedly the most expensive way to find this kind of thing out, you can imagine as the costs drop how amazing it would be to have a full picture of all the chemicals and all the microbes in your environment. And that's especially relevant now that we're starting to do this kind of mapping for COVID to understand where it goes in a hospital environment or in an isolation dorm or in one of the research buildings on campus that's inadvertently had someone who later developed COVID working in a lab. And this is all fed into our project funded by the National Institutes of Justice, using this information to build lifestyle chemistries from a swab of something like your phone or other object that you touch, where we can tell a lot about you from those objects. So the first large-scale attempt to understand the microbes on the healthy human body was the Human Microbiome Project. And so my lab was involved in this in a number of respects, but we were just part of a large 400-person consortium around the United States and internationally. And we studied about 250 people, all healthy, who were swabbed at up to 18 sites on the body and at up to 3 time points. And we collected 4.5 trillion bases of DNA in this study, so 1,500 human genome equivalents. But all about the microbial DNA rather than the human DNA. And so the great thing about this project is we had an unprecedented amount of DNA sequence data concerning the microbiome, 4.5 trillion A's, T's, G's, and C's. But that was also the terrible thing about the project. And this just gives you an idea of the challenge we face. So this is the first 0.1% of the first file of the HMP. And it's pretty hard to tell who lives where in the environment from that, right? Like you probably can't tell that's an oral sample, let alone the sequence signatures that would let us determine that. And this is increasingly a problem, not just from a research perspective, but from a clinical perspective, because I can tell you that it's your doctor's nightmare that you're going to show up in his or her office with a huge grin on your face and tell them, hey doc, I have this great news for you, which is I had my microbiome sequenced, and now I have a list of 1,000 species they found in my gut, or 1 million genes they found in my gut. And with all this data, you can tell me what's wrong with me, right? And what's your doctor even going to do? Maybe refer you to the psychiatrist colleagues who've been crazy enough to think they can do something with that data, especially in the short time you have together. And so a lot of the goal of my lab is to make it not crazy to think that you could get something out of that anymore, and to develop ways to integrate those profiles with your own profile over time, or with other people's profiles to get something out of them. So when we reprocess all of that data with the CHIME pipeline, so it stands for Quantitative Insights into Microbial Ecology, and it takes the data straight off the sequencer and it does things like alignments, phylogenetic trees, and clustering, which I'll go into a bit more detail soon. We can start to get an understanding of what's going on in these data sets. And so when we reprocess the HMP data with CHIME into a principal coordinates plot based on unifrac distances, we get this map relating all the samples to each other. And each dot on this map represents all the complexity of a microbiome read out from its DNA. And two samples, each point is a sample. 2 samples are close together on the map if they have more similarity in their evolutionary history of microbes, as read out by a metric called unifrac, developed by Kathy Lozapone, one of my first graduate students. And then they're further apart if they're more dissimilar in that evolutionary history. Now, there's no disease on this map because these are all healthy people, but when I color by the main thing going on, you can see immediately that the mouth, the gut, the skin, and the vaginal samples emerge almost like different continents on this map, where different sites on the body from the same person are completely different from one another. And to highlight this here, I'm just showing you the mouth and the gut from the first person in the HMP. But it wasn't until we did the Earth Microbiome Project, where we crowdsourced hundreds of thousands of samples from the scientific community around the world, that we truly understood how different these parts of the body were in their microbial ecology. Because what we could do is we could go out on the planet's surface and ask what 2 sites are just as different from each other as the mouth and the gut of this one individual. And if you think of your mouth as being kind of like a coral reef full of complex mineralized structures that maybe your dentist bugs you about from time to time, covered with biofilms. The amazing fact is your mouth is as far from your gut as the water in this reef is from the dirt in this prairie in terms of its microbiome ecology. We never expected that, right? That a few feet along the length of the human body could make as much of a difference to your microbes as thousands of miles across the Earth's surface. So one really important concept is that our microbiome journeys on this map matter for disease. And everyone I'm showing you on that map is healthy, but we can start to integrate people who are not healthy in these kinds of microbiome displays. And this is Larry Smarr, who's the founding director of CalIT2, and who's a very interesting character, in part because he's a leader in the quantified self movement. And shortly after I got to UC San Diego, 6 years ago now, I was explaining to Larry, like I was explaining to you, the HMP data, only instead of using a laptop, I was using the 64 million pixel display wall, the Vroom over in Atkinson Hall. And he told me, well, you know, Rob, I have a very interesting microbiome myself because I'm an IBD patient. How do I get myself onto that map? So shortly after that, this box showed up in my lab, the first of many. And Larry's very interesting because he's been releasing data about himself on the internet for the last 20 years, starting with a single dimension of his weight, and then going to dozens of dimensions with blood tests, thousands of dimensions with microbiome tests, now millions of dimensions with shotgun metagenomics. And you can see some substantial fluctuations in his weight over the period of sampling. But when he sequenced a bunch of those samples, he came up with this. And what you're looking at is a display over time of the families of bacteria in Larry's gut. And don't worry if you don't see any obvious patterns in there, because neither did Larry, despite spending some huge amount of effort and compute time on generating this display. But we got all of the data and reprocessed it with our pipeline. This was led by Justine Debelius, a grad student in the lab at the time. And what you can see is a very clear blue and red region. This is just rotating the frame around. When we animate, you see he starts off at one edge of the blue, travels directionally through the blue, crosses over into the red, and then bounces around at random in the red. So that's cool, you're probably thinking, but what does it have to do with Larry's health? And that's where we can take all Larry's health data and cross-reference it. What we can see is that the initial shift in microbiome ecology was caused by antibiotics. And then this directional period where he's having frequent IBD symptoms and losing weight, that's where he's directionally moving through the blue. He changes his medications and then moves from the blue regime into the red regime. And then in that period where he's bouncing around at random in the red, what you can see is that his weight goes back up to a healthy set point and his IBD symptoms are basically gone. And so what's exciting about this is the concept that if you have one of these personal microbiome maps, you'll be able to define healthy and unhealthy regions of the space for you. And so, so if we had known about Larry's configuration space, we could have told him that as soon as he got into this red region, he was going to be fine. And remember that all this came out of that incredibly noisy underlying taxonomy data that I showed you a few slides ago. Now, you might be thinking, well, this is all very nice if you're someone like Larry who directs a large institute, but will it expand to other people? And so we worked with Janet Jansen to expand this to a larger cohort. And what you can see, I want you to focus on the red and the yellow traces, which are ileal Crohn's patients. And what you can see is that those lines are very long as we animate the trajectories over time, whereas the healthy controls in green especially are restricted to a very small region of the configuration space. And so what you can see is that the people with ileal Crohn's especially are changing a lot more, and they're exploring completely different regions of configuration space than the people who are relatively healthy. And so Yoshihi Vázquez-Beza, who was a graduate student in my lab at the time and is now the director for software integration at the Center for Microbiome Innovation, was able to model this as a healthy plane, looking at the distance of each point from the plane. And that geometric model, based on the unifrac distances reduced by pico-A, gave us better disease classification than calprotectin, which is currently the gold standard biochemical assay for telling whether someone has inflammation associated with Crohn's or not. So this is very exciting because it suggests that out of all that complexity and noise in the data, we can come up with a very clear signal based not on the based on the static snapshot dynamic. Now, the human microbiome is very complicated and challenging to understand. And so this is why we often run up against the limits of human intelligence and have to turn to artificial intelligence to really understand what's going on. And a few years ago, so back in 2017, our Chancellor and John Kelly III, one of the senior VPs at IBM, created the Artificial Intelligence for Healthy Living Center that I co-direct at UCSD with Dilip Jesti, who's in the Stein Institute for Research in Aging. And this is part of the AI Horizons Network. And essentially what we're doing is we're using AI to understand how people age successfully versus unsuccessfully and what role the microbiome plays in this. So one of the big successes from the center was this paper published in M Systems, where we found out that we can tell your age from your microbiome, where the skin, the mouth, and the gut microbiomes can all predict chronological age using random forest models. And in AIHL, we're taking a two-pronged approach, where with one thrust that I lead focused on the microbiome, and a second thrust that Dilip leads focused on healthy aging. So one of the key questions that we have at the moment is, how does your microbiome age compare to normal? So the idea is that maybe you'd expect to see a direct linear relationship between your chronological age and your microbiome age. And then what we're interested in is people whose microbiomes look especially young for their chronological age, who we suspect are going to be especially healthy, and then those who look especially old, whose microbiomes look especially old for their chronological age, and those are the people who we suspect are going to be especially unhealthy. And to do this, we rely on a database called Cheetah. This is published now in Nature Methods, but now spans hundreds of thousands of samples from around the world. And this is just showing you a map of all those samples. So this is a large infrastructure that we've created here at UCSD, where all of the public data and reference sequences can be downloaded by anybody. And a lot of this is based on ribosomal RNA, which is useful because it's universal. It has slow and fast evolving regions that can be used to pull out the right parts of the molecule in the lab, and then identify the parts that are unique to particular kinds of taxa. And we have massive databases of known sequences we can use. And this was pioneered by Norm Pace, who used ribosomal RNA to define the overall tree of life with Carl Woese. And so just to give you an idea of this, Darwin came up with the original concept of the tree of life in 1837. In 1977, trees could be built with 13 taxa using molecular methods. But recently, as part of this project, we created a new tree with over 10,000 complete genomes using 381 markers, collaborating with Siavash Mirarab in ECE, using a method he introduced called Astral to do that consensus tree. And just to give you an idea of how big this new phylogenetic tree is, we covered 1.16 trillion amino acids over 10,575 genomes by 381 genes, using a combination of Astral and RAxML on Comet, one of the major supercomputers at SDSC. And one thing that was interesting about this is that we really redefined how the tree of life looks. We are looking at these global markers. What we saw is that the archaea, the eubacteria, and the so-called CPR, the candidate phylum resolution, candidate phylum radiation, have about equal taxonomic status. Whereas if you just look at the ribosome, either the RNA or the proteins associated with it, that's where you see the archaea separating from the CPR and the bacteria in a different way. And to build this tree really pushed the limits of computational power. So like I said, we built it on Comet, we used a total of 2.3 million CPU hours on Comet to build the tree. So one really important aspect of this improved tree is that having a better picture of how life is related to each other allows us to do things like the Unifrac algorithm better and improves age prediction. And so what we found is that not only could we improve the age prediction overall from marker data, but by looking at genome resolved and strain resolved data, we could substantially reduce the average error. So the average error in prediction of age from an individual. And this was a really important advance because prior methods using shotgun metagenomics had typically provided worse predictions than methods using the 16S ribosomal RNA marker gene. And so we're further improving this in collaboration with IBM using their Auto AI tools at the moment. Now, switching gears from age to cancer, another important topic that we're tackling by way of the microbiome is the problem of cancer. And this is primarily the work of Greg Poor, who's a very talented MD PhD student in the lab. But typically cancer is viewed as a sterile entity, and the hallmarks of cancer papers from Bob Weinberg have now been cited over 75,000 times, but they don't mention microbes at all. And Greg found out that all of this work is missing an important dimension. So, for example, if you look at colorectal cancer, there have been some intriguing hints that cancer is not sterile. And so, for example, these are electron micrographs of colon cancer cells, and every arrow is a bacterium that's found in those cells. And in fact, bacteria are found in 40% of patient biopsies from people with colorectal cancer. So Greg performed the largest scale proof of concept that cancer is not sterile using TCGA, which is the Cancer Genome Atlas, and it spans over 2.5 petabytes of data. So that'll take 212,000 DVDs to store based on data from 11,000 patients. And here's Greg grinning very proudly because his paper was published in Nature earlier this year. And essentially what Greg did is subsetted the relevant data to 590 terabytes, then aligned it to human reference genomes, threw all that stuff away, and then took the sequences that did not align to the human reference, put it through a custom pipeline, including data transformation that includes batch correcting algorithms that I'll describe a little more later, and then using machine learning and Bayesian source tracking to understand where the sources of different microbes are and what microbes provide signatures that specifically discriminate among different cancer types. So one thing that was cool about Greg's analysis is that it revealed significant amounts of microbial DNA in most tumors. And so what you can see is that typically between 1% and 4% of the DNA in the sample comes from microbes of some kind. And then from about 0.1 to 2% of those microbes, we can identify to a particular microbial genus, although many of the reads are unidentified at present. And one big problem with using TCGA data is batch effects. So on the left, you can see the uncorrected data from TCGA, where there's a very large effect of what sequencing center did the work. And Greg used an algorithm called VOOM to batch correct the data. And what you can see on the right is that after batch correction, the data is much more centered without that sequencing center effect. And what we found is that this dramatically increased the fraction of the variance attributed to biological variables as opposed to technical variables. So this is just a picture of Greg's machine learning framework, including fourfold cross-validation, a gradient boosting a machine architecture, and then the final tuned model was used to do class prediction on testing data that had been left out of the training data set, and then used to produce ROC curves, PR curves, confusion matrices, all the rest of the stuff that you'd expect. And this gives you a sense of the classifier accuracy that Greg was able to achieve, where what you can see is that typically the ROCs are very nice, with the area under the curve being in excess of 99%. And what this means is that we have very accurate ability to classify a subject as either having a cancer or not having a cancer, and also understanding which cancer it is, whether we use the DNA or the RNA. So Greg's had enormous success with us, and beyond the Nature paper, he started up a company called Micronoma to commercialize this technology and get it into the clinic as a diagnostic tool. Now, everything I've told you about so far has been based on sequences, but sequences are just one part of the puzzle. And what we really need to understand is how those sequences fold into structures, how those structures provide functions, how the molecules interact with each other, and then how those interactions can be facilitated or disrupted by small molecule drugs that can act as therapies. And in this respect, the recent announcement that AlphaFold 2 was giving much better prediction accuracy is very exciting, and we're currently looking at incorporating that into our structure prediction pipeline. We've been engaged in this project called the Microbiome Immunity Project with researchers at the Broad, at the Flatiron Institute, and with IBM's World Community Grid. And here's just a photo of the team taken a couple of years ago. So what we're trying to address here is how much of the structure space in the human microbiome is unknown. And what you can see here is that although sequence data like UniProt is growing spectacularly, the PDB, which contains all the structures, is growing slowly because experimentally determining the structures is very time-consuming and difficult. And so the problem is that each protein sequence, it can take about 1 CPU hour per domain, and there's 10 to 20,000 domains, and then at the end of that, 10 to 20,000 trials that you have to do to get a convergent structure, and at the end of that, what you get is one structure out of it. And so the solution is to split up all that work massively parallel over the world community grid, where we're using spare cycles on many people's computers to do it, then aggregating the structural information at the end together with Rich Bonneau at the Fight Iron. So the World Community Grid is a philanthropic initiative of IBM corporate citizenship, and currently over 750,000 individuals and 460 organizations are contributing compute to this. And that's what we're using for this project. And just to give you an idea of the progress, we started the project in August 2017, and we've already produced more data than the entire PDB with these structural predictions. And you can see that we've consumed many CPU centuries of time on this project. So where we're going with this is also to be able to do improved structure-based prediction of function using graph CNNs. And essentially what we're trying to do here is we're trying to take the structures, including the predicted structures, and from the structures understand what the function is by building a network representation of the residues and understanding where the active sites are. And so we're using a combination of LSTMs and graph CNNs to do this. And one thing that's been very exciting about this has been that we can get high quality function predictions even with the de novo models. So, so the models that we're producing with computational folding are giving us input that's almost as good as what we see with bond angles and distances that are derived from techniques like X-ray crystallography and NMR. And so what's very exciting about this, especially with the ability to incorporate AlphaFold 2 predictions into this, which will dramatically expand the range of protein space we can access. What's very exciting about this is that we can get better joint predictions of the structure and the function from each of these proteins in the microbiome. And we've already been able to get it to 4 CPU hours rather than tens of thousands of CPU hours per structure, validated against the CPU centuries of work we did on World Community Grid. And so one thing that's cool about this is we've already been able to discover new folds that have important functions, like completely new classes of fatty acid binding proteins that had not been seen before, but that we could verify the functions of through this process. Coming back to microbiome sequences, all of this kind of microbiome work has led to a lot of excitement in the general public. And back in 2012, when we published the first results from the Human Microbiome Project, a big issue was that there was no way for ordinary members of the public to get involved because it was a large bureaucratic government project and you had to meet a lot of exclusion criteria and happen to be at one of the recruitment sites to be able to get involved in this. So, so we turned to crowdsourcing and crowdfunding to basically allow anyone who was interested to get involved in the project by supporting the cost of adding them to the project, much like National Geographic's Genographic project. Of course, not everyone wanted to know what was in there, and these are middle schoolers being told by Daniel McDonald, the scientific lead of the project, about how we're going to use robots and lasers to read out the DNA of the bacteria in their poop. You can see a certain amount of response heterogeneity there. We don't yet know if that's linked to the microbiome, but sooner or later we'll have the sample size to find out. But basically, relative to other citizen science, crowdsourced and crowdfunded projects, this is very large. So we've raised over $2.5 million. And in the initial publication, we looked at over 15,000 samples. We're up over 25,000 samples now, although it's been on hold during COVID. With the sample size, we can see relationships between the microbiome and things you might not expect. So especially from what I told you earlier, you're probably not surprised that the microbiome changes as you age, but you might not have expected that it changes depending on how many hours of sleep you get at night, which you might not have expected it was connected to. And by having all of these people, we can subsample from them and ask how many people per group do you need to see a particular effect. So this is the number of observations per group on the x-axis and statistical power on the y-axis, where you can see for something like age or IBD or collection season or other curves on the left, you need a few dozen people per group to tell that apart. Whereas for something like whether you're male or female, how much you sleep, how fat you are, you need a few hundred people per group to tell that apart. But one thing that's exciting is that the steepest curve on this graph is the number of types of plants you eat, which is an unexpected dietary factor that you have complete control over and can give you a substantially more diverse microbiome. In American Gut and its international umbrella organization, the MicroZetta Initiative, where we're using gut microbes as a Rosetta Stone to understand diet and health and their interactions, we've rapidly been retooling the project to read out RNA rather than DNA, so we can look at the SARS-CoV-2 virus, which doesn't have any DNA. And we've already been working with a number of different cohorts to get it out rapidly to people that we have a good understanding of. And this is Edgar Diaz, who's the project manager for American Gut. This gives you an idea of the pallets of shipping containers of kits. The pallets of kits that are stacking up to get delivered. And so we've added a COVID-19 questionnaire to the American Gut Project based on standardized questionnaires from NIH and other sources. We've also put together a detailed set of instructions, including additional collections like blood for serology and so forth. And like I mentioned, we're doing finger stick collections of blood for serology. We're doing saliva collection and then nasal and fecal swabs. And this had to be approved by a lot of organizations, including the FDA, FedEx, our local IRB, the IATA, so we could transport them internationally, all kinds of stuff like that. And one thing that's been exciting, and this has been led by Cameron Martino, who's a bioinformatics and systems biology grad student in the lab, is taking those age prediction models and connecting them to SARS-CoV-2. So COVID-19 affects the elderly a lot more than it affects younger people. And Cameron was curious whether this could be linked to age-related changes in the microbiome. And so what he was able to find was big changes in heparin sulfate metabolism, which is one of the surface receptors that the SARS-CoV-2 virus binds that systematically change with age. And he was able to look at different populations, including FinRisk, which is a set of 7,500 people from Finland who provided a fecal sample for science back in 2002. And we have all their health records going forward. Also people in the American Gut Project, where you can see that the heparin sulfate modifying genes decrease substantially with increased age in these different populations. So this gives you an idea of how it all ties together and can help us respond to urgent threats like the current pandemic situation. So you might wonder, what's our vision with all this? And what we really want to do is we want to make this stuff useful at the individual level and rapid so that you can draw conclusions from it. And this map is just to reorient you on the HMP data with the mouth, the skin, the vaginal, and the fecal communities. And the best example we have of why you care where you are on this map is C. diff, so a particularly nasty form of hospital-acquired diarrhea. When I show you the microbiomes, these are stool microbiomes of C. diff patients, but you can see those orange spiky shapes are nowhere near the healthy fecal communities down the bottom in brown. And what's going to happen is 4 of those patients are going to get what's called a fecal transplant from one healthy donor. This is work we did with Alex Karutz and Mike Sadowski at the University of Minnesota a few years ago. And if you're wondering what a fecal transplant is, this is Bill Sanborn, who's our chair of GI, about to deliver his first one using hospital-grade stool that he got from a nonprofit called OpenBiome, because you have to remember that the FDA regulates stool as a drug because it's a substance that's been shown to have an effect on clinical endpoints. So anyway, 4 of these patients are going to get a stool sample from one healthy donor who, as you can see, was in the healthy region in brown. And you might wonder, are we even going to see anything on the scale of the whole microbiome, given what I've told you about its diversity and its complexity? And each frame in this animation, which again was put together by Yoshiki, is 1 day in the lives of these developing microbiomes as they're under the influence of fecal transplant. And what you can see is for all 4 patients who got the fecal transplant, immediately, so in just like 2 or 3 days, they reset to the healthy state, and then they stay there for months and months of follow-up. And this is amazing because their microbiome ecology is completely resetting just with a few grams of stool. And coupled to this, all their clinical symptoms are gone. And fecal transplant is 95% effective for C. diff in many different studies now. And the American Gastroenterological Association is now recommending it as a first-line therapy rather than a therapy of last resort if you have C. diff. And so it's been really exciting to help contribute to those kinds of recommendations in that research. But the broader question that we have is, for what other diseases can we identify a problem with patients, whether it's something obvious like you're seeing here in C. diff, or something much more subtle like an obesity, and then guide it back into health, into the healthy microbiome state. And so what we want to do is to do this for all these different diseases that have now been linked to the microbiome, including things like IBD and IBS, but also more surprising things like cancer, like heart disease, like autism, like Parkinson's, even Alzheimer's we're working on at the moment. And so what we need to do is we need to find the good and the bad places on the map, but we also need to develop a kind of microbiome GPS that tells you what to do to get from the bad place to the good place, whether that's something that seems as extreme as fecal transplant or something that seems as gentle as diet, which has a huge impact on your gut microbiome over the longer term, or something in between like phage therapy, which we're doing here at UC San Diego, drugs, probiotics, prebiotics, and so forth. And so ultimately, we want to develop this kind of system and make it so easy to use that even our children can use it. So you could imagine a smart toilet where as soon as you flush, you get an instant readout of the microbiome that's delivered to your smartphone, which, let's face it, I bet you're using in there anyway. And when my daughter was 3, when we took this photo, she'd already learned not just how to use a smartphone, but where we all too often use them. And at that time, it would be too slow to run the stuff on a phone, but today we can. And for example, this is her own microbiome developing from an undifferentiated state towards the adult state defined by American gut into those different body sites over the first 3 years or so of her life. And this is the kind of thing you can now explain to a motivated 9-year-old, which she is currently. It'd be really with time and patience. And the idea is that you could explain it in the rush context of a doctor's visit to a busy patient and a busy clinician in a shorter time with further improvements. But we need to be able to do this in real time. And the studies that I showed you, mostly we analyzed the data years after the samples were collected. And that's kind of like trying to troubleshoot a self-driving car just by analyzing the logs after it crashes. We need to get the information in real time so that you can see how your microbiome is doing and how to guide it. And so our vision with this is to get the stuff out of the research lab and into a consumer setting. So you could imagine that as you examine yourself in that mirror in the morning, maybe as you breathe on the mirror, your breath is whisked away into some kind of instant chemical analysis by mass spec or NMR or Raman spectroscopy or something, and you get a near real-time chemical readout. Obviously, we're trying to make it more user-friendly than this, because unless you really loved organic chemistry, you probably don't want to see this thing first thing in the morning. But then the idea is to translate that into a microbiome profile like you would get from American Gut, and ultimately to put yourself on the microbiome map where we could identify what you're at risk of, and then either tell you to see a doctor about that, or tell you things that you could take into your own hands as a consumer that would mitigate your risk. And then, obviously this is the science fiction part of the talk, but the idea is that your smart mirror could then communicate with your smartphone over IoT. And then when you're standing in the supermarket and you're confronted with 1,000 kinds of yogurt, you could use augmented reality to zoom in on the product that has the microbe or the molecules you're looking for, and then scan the barcode to prove that you got it. And you could also imagine having your smartphone communicate with your mirror about what you did during the day that affects your microbiome. So what you ate, who you were with, how many minutes you spent outdoors, which has a substantial impact, how much exercise you got, all these different things. And then use that to modify your aging profile in the mirror to show you visions of yourself 5, 10, 20 years down the track if you behave every day like you did today, or what you might look like if you did a little better or a little worse. So that's the kind of crazy stuff we're up to at the Center for Microbiome Innovation that I direct, where together with an expanding range of corporate partners and over 140 UCSD faculty, we're trying to do a number of things as crazy as this in health, in food, and agriculture, in various industrial settings, to understand how to not just know a bunch of stuff about the microbiome in an ivory tower, but how to modify it to do things that are useful. And as you've seen through this talk, bioinformatics and AI play a lot of key roles in that. So with that, I'd like to thank briefly the many people in my lab, quite a few of whom I mentioned during the talk, but a lot of whom work on other topics that I didn't cover here. Literally over 1,000 collaborators around the world, our many different sources of funding, including the tens of thousands of members of the public, where we're especially grateful for their contributions to the American Gut Project. And finally, thanks again for inviting me to do this talk. I'd be delighted to answer any questions you have, or feel free to email me if we don't get to your question in the Q&A. And also, I cover this material more in my two books, Follow Your Gut and Dirt is Good. So with that, thank you. I'll just stop the screen sharing and look in the chat. And happy to answer any questions you have. Thank you.
Okay, so if you have any questions, just either text in the chat or raise your hand and I'll ask you to unmute.
Yeah, and I'll just stick my email address in the chat if we don't get to your question during the Q&A. I guess it got dark while I was doing that, so let me just turn the light on so you can see me again. Always the challenge with the 4 p.m. time slots at this time of year. Anyway, questions? Oh, I should say that we have a lot of openings in the lab at the moment for undergraduate research, and undergrads have contributed a lot to many different microbiome projects in the lab over the years. And we've got a lot of compelling needs at the moment, everything from fundamental algorithm development to things like putting together improved dashboards so we can rapidly report COVID results to public schools or to UCSD leadership or that kind of thing. So there's a lot of opportunities to get involved, either in microbiome research generally or in addressing the current pandemic.
So I guess I have a question. So you were talking about using graph neural networks to predict like protein function and folding. I was wondering what your thoughts on AlphaFold. They recently came out and said that they were able to have their deep neural network like predict folding. I was wondering what your thoughts on that were.
Yeah, it looks awesome. So the recent one is the second version of AlphaFold, and the publicity is that that the Casp assessment was just released. So this is an international consortium to assess different protein folding methods in an unbiased way. And AlphaFold 1 came out 2 years ago, and we had already been doing a lot of validation of Rosetta, which we've been using, versus AlphaFold 1, which is a lot faster. AlphaFold 2 is a lot more accurate and also handles transmembrane proteins, which is a major advance because they're very difficult to crystallize. So it's been very limiting to do X-ray crystallography of them. And so I think AlphaFold 2 is going to be completely revolutionary as long as it works in the hands of everybody rather than just the team that developed it. One thing that's often a problem with industry projects is that the transparency that you have and want in scientific work isn't always there, and there may be extensive tuning that's not available to the community. So sometimes it's been quite difficult to reproduce the results. But in terms of just being able to always and automatically get a structure for every protein you have a sequence for, that would just be transformative and revolutionary, in the same way that being able to look up a sequence in GenBank rather than having to figure it out from scratch was absolutely revolutionary. Let's see. So a couple of questions. So Christoph, thanks for your kind comments. Sorry, I'm really sorry to hear about your experiences with Levaquin. The evidence base on FMT for post-antibiotic issues is very thin at the moment, so it's something that's only happening in research projects, not something that's clinically recommended, but it's certainly something that there's a lot of interest in, as is trying to understand why some people are resilient to antibiotics and other people are much more severely affected by them. And that's exactly the type of research that's going on currently. And then that ties into the next question. What do I see as the future for antibiotics? I think the future for antibiotics is that we're going to have a much better understanding of when to use them and when not to use them. So the way we treat antibiotics at the moment, I think, is like the way that radium was treated in the 1930s, where everyone was so excited about this material that you could use to make watchdials glow and so forth. And people would do things like paint their entire basement with radium paint without worrying about, was having your whole basement glow may be bad for other reasons, like it's going to make you more radioactive and your bones will all disintegrate. So, so I think in the same way that we're now much more careful about how we use radiation and how its medical and industrial uses are very circumscribed, and with a lot of regulatory oversight. I think antibiotics are going to go the same way, where sure they're life-saving, but we need to be a lot more careful about their harmful consequences. And so understanding what's the right antibiotic for what patient, and being able to do the sequencing rapidly so that we can make those decisions early on, that's going to be very important. And this large-scale project with Rady Children's Hospital at the moment, trying to get off the ground to do this in a pediatric context with critically ill children, with Steve Kingsmore, who runs the Rady Genomics Institute. And he's mostly focused on human germline sequencing, but he's getting very interested in the microbiome. Let's see, so my opinion on the importance of understanding the relationship between biochemical pathways and the microbiome, I think it's critical. And a large part of the reason why I moved here was so I could collaborate closely with Peter Dorrestein and Mo Jain, who were able to read out metabolomics in very high throughput from a microbiome sample or from a blood sample, respectively, and with Bernard Paulson, who's a leader in genome scale metabolic reconstructions, where basically from a genome, Bernard is able to build a network of the stoichiometry of all the reactions that you would expect there. And Bernard's former trainee, Ines Thiele, as part of the Alzheimer's project that I mentioned briefly, where we're trying to understand how the microbiome contributes to Alzheimer's. But Ines is doing some awesome work doing a full-scale metabolic model for humans and also for microbes that could be combined to get a sense of human microbial co-metabolism. And we're looking at how that corresponds to development of Alzheimer's, and how that can be affected by different dietary trials like the MIND and the DASH diets. So what's very exciting about this is being able to integrate metabolism with what we know about genetics. It's really the direction the field's going, so that we can establish causality rather than just discovering patterns, and so that we can understand in detail how to not just read out what's happening, but to modify what's happening in a desired direction. I may be biased in this though, because my formal training, like my undergraduate degrees in biochemistry, so I guess having that background in biochemical pathways has been very useful for subsequently interpreting microbiome data as I've shifted research into that area over the last 2 decades. Let's see, what does the microbiome of the human respiratory system look like, and how does it interact with COVID-19? So the second question, great question, no data that I'm aware of. Most of the data that we have so far is looking at the gut microbiome and its relationship with COVID via interaction with glycans and the ACE2 receptor. The microbiome of the human respiratory system, basically the nasal microbiome looks like the skin, and the throat microbiome looks more or less like the oral microbiome. So although the HMP initially looked at what they called airway samples from the nose and from the throat, ultimately we had to reclassify those with the skin and with the with the oral samples respectively to get to let the data tell us what the clustering pattern looked like. And then I was involved on the steering committee of the Lung HIV Microbiome Project, which NHLBI ran for 5 years in the, let's see, starting around 2012, maybe 2011. The big problem there is that it's really difficult to collect DNA from the lungs without contaminating it with DNA from the airways. And what we think now is that in healthy people, the lungs are probably sterile, although there are traces of dead bacteria that you'll find in there and read out from the DNA. But basically, your lungs are covered with antimicrobial peptides that are very good at killing anything that lands there. Do I see this microbiome research having impacts on agriculture and food production? Yes, absolutely. So I didn't talk about this much here, but the soil microbiome, animal microbiomes, plant microbiomes are huge topics where there's a tremendous amount of activity at the moment. And the FAO, so the UN's Food and Agriculture Organization, is doing a lot of investigation at the moment on the links between the microbiome, soil health, crop health, animal health, and how we can improve the food system overall to be healthier both for us and for the planet. So in that respect, one thing that was fascinating is David Tillman at the University of Minnesota, one of the world's most famous ecologists, and some of his colleagues published a paper in PNAS last year where they looked at whether there was a trade-off between foods that are good for you as an individual and foods that are good for the environment. What they found was that rather than being a trade-off, there was a very strong positive correlation where foods that are bad for people are also bad for the environment, with red meat being the poster child for that, obviously. And then foods that are good for people are also good for the environment in terms of having relatively low inputs. So that's things like whole grains, legumes, and so forth. So there's a lot of interest there. And then in terms of pesticide use, there's been a lot of work on how you can use particular microbes to combat fungi, to combat insects, to combat nematodes, to combat bacterial and viral pathogens. And one thing that's been interesting there is modifying the microbiomes that are intracellular within the plant to help with disease resistance, and also with things like salt tolerance and drought tolerance. So it's a very large area with very active research going on in there. What do we do to normalize the data being analyzed, and how do we identify potential contaminants? So I didn't talk a lot about the lab processes, but we run a lot of technical controls, both positive and negative controls on each plate, where essentially we sequence samples that don't have any of the biological specimen in them to see what comes along for the ride when we do the lab processing. And we also focus a lot on methods for understanding well-to-well contamination in the robotic liquid handling infrastructure, potential sources of contamination in the reagents by sequencing reagent banks of various kinds and so on. In terms of data normalization, the important thing to understand about microbiome data is it mostly lies in the simplex rather than Euclidean space, because we analyze it as compositional data, and so using methods like ANCOM and Songbird that explicitly take into account the compositionality is really important. And then for the cancer data that I showed you from Greg Paul's work, that was using VUM for normalization. Let's see. Are there any other approaches to manipulate the microbiome besides FMT? Yes, so there's a lot of other approaches, including drugs that target the microbiome, including but not limited to antibiotics. So, for example, metformin has a big impact on the microbiome. There are what are called prebiotics, which are compounds that feed particular kinds of bacteria that are good. So, for example, using inulin to increase the amount of butyrate fermentation in the gut. There's phage therapy, where you use a virus that affects bacteria to kill particular types of bacteria that you don't want. There's small molecule inhibitors of microbial enzymes. So if a microbe is doing a biochemical reaction that you don't want, instead of drugging an enzyme in the person, you can drug an enzyme in the bacteria to wipe it out. You can introduce individual bacterial strains or cocktails of strains as what are called live biotherapeutic agents. And for example, Jeff Hasty, who's in bioengineering and biological sciences here at UC San Diego, is engineering bacteria that can deliver themselves to tumor cells and then have a lysis circuit where when they build up to a particular population size, they burst. And so what that means is that whatever the bacteria are producing, like a small molecule anti-cancer drug, you don't have to get it out of the bacterial cell because that's handled by lysis. So that's in clinical trials at the moment as a delivery mechanism for anti-cancer proteins or other agents that are hard to deliver into the tumor. And then diet for modifying the microbiome is proving incredibly valuable. So we've been engaged in some work with a number of different investigators looking at how you can reshape the microbiome with diet and its effect on inflammatory diseases like rheumatoid arthritis, IBD, and so forth. What's interesting there is that there's very high individual variation in response. And so if we can link that individual variation to the microbiome and remove it, so in other words, tell people what drug or what diet is going to work for them personally, that'll have a huge impact because at the moment, individual variability limits the effectiveness of a lot of therapies. And if we could identify a subset of people that the therapy is going to work amazingly well for and tell those people to take it and tell everyone else not to take it, that's where we're going to have a much bigger impact on some of these difficult to access conditions. All right. Well, I realize we're a bit over time, and I know you're all busy with your end of quarter assignments and that kind of thing. So if you have additional questions, feel free to email me. My address is just robknight@UCSD, and I put that in the chat. And especially if you're interested in research opportunities, either in the microbiome or related to COVID-19 and helping our campus and our community address the pandemic, please do get in touch. There's a lot of exciting stuff to do. Thank you.
Thank you for giving us talk, Dr. Knight. We really appreciate it.
Absolutely. Thanks again for the invitation.